So we will be discussing about little bit about my company, what I work, what we do, and then with the help of few case studies and few um, research things, we will get to know how can a computer science student can also be fit into this kind of ecosystem. So ma'am, shall I continue?
Yes. Please.
Okay, I think there is some kind of. Okay, I
Yeah,
don't need it. Okay. First I'll start with my present organization. It's a MiRXES Labs. It's a Singapore headquartered uh, company. So MiRXES is basically we have a headquarter in Singapore. We primarily work with the RNA technology, and we are into the area of diagnosis and prognosis um, with the cancer and other infectious diseases also. So our R&D is in Singapore, our R&D is in China also, and also our production is in Singapore. MiRXES is not only one company; it's basically group of many companies or many kind of organization which are into the healthcare system. So MiRXES is also own M Diagnostics. MiRXES also owns the MiRXES Genomics. MiRXES also have a clinic which is called Early, and MiRXES has own developing and R&D and production facility related to the in the field of diagnosis. So I will start with the basics. As per my knowledge, you all are from the most of the students are from computer science background, like mathematics background. So they have a little bit idea or no idea about the biology of the things. So I'll start with very basic so that you can also understand how things are being done into a biotechnology company where a doctor can meet with a mm, developer or a programmer. So we have offices and services. We provide our services in more than 40 countries now, but we have offices in China, in USA. We have office in Japan, in Vietnam, in Malaysia, and also in many other Southeast Asian countries like Indonesia. Also, we have. Uh, Unfortunately, in India, we do not have any office, but also we have some people who are working from India just to market our products and all. But the main facility, main manufacturing and R&D, it's in Singapore only. So I work from Singapore office. If I talk about the product and services of MiRXES, first and the most important thing, we provide the sequencing services. Also, during this talk, I'll explain you what is sequencing, how we do it, and what's we offer to our clients. So this is like business to business thing. We have several clients for whom we do the sequencing services. Then we have our hero product that is called a GastroClear. GastroClear is basically a cancer diagnosis kit using blood sample. So it takes the plasma sample as an input and gives you the result of GastroClear. This is, I would say, it's a hero product and the most popular product by the MiRXES. Then we have one more treatment kit for the GastroClear only, gastric cancer only. It's by M Diagnostics. So these are few of the product and services. Apart from these major services. We are into the solution of the infectious disease also. So if I talk about the infectious disease, so swab collection, maybe the plasma collection, RNA extraction, saliva collection kits. So all these kind of kits also we manufacture, and apart from that, we also manufacture the RNA extraction instrument. Apart from that, during COVID, we also generated the PCR kit. In India, we used to we know them by name RT-PCR. So we also produce RT-PCR kits, and also we produce the rapid antigen kit for the cancer diagnosis, and including India, we supply to many countries during the COVID. So in Singapore, we are the most developed RT-PCR kit. Like we have developed the most many of the RT-PCR kits in collaboration with the government in Singapore, in Malaysia, in Indonesia. So we have collaboration with different government also related to the RT-PCR kits manufacturing. So these are some of the work we have done or we are doing. Um, so I'll continue with the basic understanding of our work. So I'll play this video. I hope you understand a bit about the technology. So I'll tell you, in the blood, in the saliva, in the urine, in other body fluids, uh, we have RNAs into it, and in the RNA we have multiple microRNAs. And these microRNAs, the research have already discovered that those microRNAs and some part of the DNA also is associate with the chronic diseases like cancer, like diabetes. So, having this thought in mind that the microRNA can be a breakthrough uh, uh, thing in the cancer discovery, like early cancer discovery, this whole MiRXES is being started. So if I explain you a little bit about the simple steps in the diagnosis system using the liquid biopsy is first the blood sample or any other fluid sample, maybe the urine sample, is being taken from the patient, and then we perform some kind of pre-processing into the that fluid. And the sequencing is also part of this kind of pre-processing, because after doing the sequencing only, we will be able to extract the DNA and RNA from the fluids. So apart from the sequencing, this process also included the mutations of those fluid, that from even from the very small amount of the fluid we can generate so many microRNAs just to experiment and just to identify the disease. So this is the first step. And once this step is being done, then we come to the main point, which is related to the statistical analysis and data science thing. On that particular data, we apply some kind of AI techniques and machine learning techniques, and after that we can predict the disease, whether it's a tumor is cancerous or tumor is non-cancerous. So in addition to this, this diagnostic system can also be extended to to the treatment or to the prognosis system only. So we do the biopsy, we consider the microRNAs. From those microRNAs, we can also check the what kind of drug will work on the patient, and based upon the statistical analysis, we can provide, or we can do the appropriate treatment according to the diagnosis. So this is like a whole a circular system from diagnostic to treatment using any liquid biopsy. So I'll try to explain you about this new technology with the help of already established technology, which is the tissue biopsy. So I'll explain you a bit. If any person have uh, lung cancer, so what doctors do, doctors reach to the lung area, reach to the affected area of the lung where tumor is, and take a little bit of tissue sample from the tumor part, and then they start doing analysis on that particular tissue part, whether that tissue is cancerous or not. This is very important part in the diagnostic thing, because in generally 90% of the tumors are non-cancerous, only 10% of the tumors are cancerous. So if it's cancerous, then treatment will be given accordingly, and if it's non-cancerous, then treatment will be given accordingly. But the point is, uh, in so many cancers, this tissue biopsy is so painful. It's very painful technique, and also this technique is dependent on the expertise of the doctor, whosoever is the medical practitioner. It also depends on his expertise. Also, we need sophisticated instrument to perform these kind of tissue biopsy, especially when it's inside the organ. In maybe especially in the brain cancer, biopsy is not possible or very difficult. Even for the case of gastric cancer, lung cancer, um, and these kind of tissue biopsies are very difficult. So we have an alternate of performing the liquid biopsy. Liquid biopsy, we consider the fluid liquids in the body, maybe the saliva, maybe the blood, plasma, and then we extract the DNA, RNA from it, and from the DNA, we try to identify the disease, try to identify the cancer or any other chronic disease. So this is the whole concept of a liquid biopsy as compared to the tissue biopsy. It's way cheaper, and also it's less painful. In addition to this, it doesn't need any specific expertise to consider the samples. So uh, if I now you know what is the liquid biopsy and why we perform it. I'll quickly uh, uh, cover this portion just to come to the main computer science part, where as an computer science engineer or a developer you can, or a data scientist, as a data scientist you can contribute. So sequencing is like when we take the liquid sample and perform certain kind of experiment into into that particular fluid and generate a sequence out of this. This whole process is called sequencing. And in that sequence, this is that those sequence are the genetic sequence, our gene sequence, and that gene sequence only tells us about the disease. So after getting the liquid, the blood or the plasma, we extract the DNA out of it. From DNA, we perform PCR. You must heard about the PCR. PCR is polymer chain reaction. So in COVID test also we do the PCR on the saliva. So PCR means a polymer chain reaction. So we generate from a very small amount of sample, we generate a huge amount of sample, and then we do the sequencing into it, so that a large amount of data can be generated, large amount of genetic data is can be generated from the sample. So I must tell you, from one sample only, from like maybe the 5 ml of plasma sample only, we can generate near about 20 to 30 GB of data, of the genetic data. Now, from this huge data, this data is related to the multiple cells, related to the multiple microRNAs, multiple DNAs. Now the main work comes into the picture, where we apply computer expertise, we apply the AI into it, and we perform extensive ML into it, so that we can diagnose the cancer from the sequence data. So this sequencing, up to the sequencing, it's one part. After the sequencing, it's another part of the whole research work done by us. So as I explained earlier also, we take the real-time liquid, for an example, let's suppose blood, and from that blood we extract CTCs, we extract ctDNA, we extract exosomes, we extract other information only, and then we start performing, start doing experiment onto these things. Like we cannot directly apply the analysis part into the fluids. So we extract the information from it, then we start doing the experiment. So in simple terms, we have fluid, we extract the DNA from it, we do some kind of pre-processing and DNA fragmentation. After doing DNA fragmentation or DNA cleaning, we perform the DNA sequencing part into it. Once DNA sequencing has been done, now we just apply the classification part into it, and this classification part gives us the information about the cancerous, non-cancerous, healthy, diabetic, non-diabetic, or maybe any other disease, like maybe the COVID positive or COVID negative, any other thing. So this is the whole process of this. In case of saliva sequencing, for COVID diagnosis, we use it. It's a easy process, because number of DNAs, not so many number of DNAs are involved into it. But when we talk about cancer, as in video also it's mentioned, it's about 2500 RNAs are involved into the cancer. So it's a big uh, data to be handled for the cancer diagnosis. So I will start. I think up to this point you have a bit idea about the diagnosis and what exactly we do. So if you have any question or anything up to here, you can ask me. So if you don't have any question, you can send me the uh, uh, messages also. So I will take consider as a no, and I can continue. So the first case study I am going to present about the lung cancer diagnostics. It's a very simple thing. We just extracted the microRNAs from the uh, plasma sample, and then we perform the heat map clustering into it. It's a non-supervised kind of application, And in the first phase, we just cluster our data into the cancerous cell and non-cancerous, or the lung cancer or non-lung cancer cells. It seems like this. So in columns, we have different cells, and in rows, we have different microRNAs. So like from each and every cell, so many microRNAs are extracted, and out of these microRNAs, only one microRNAs have the significant effect into the cancer diagnosis. Like these green point have value 5 point, more than like in between of 2.55 to 5.12. It means that any microRNA correspond to any particular cell have this green value is more related to the cancer. And if it is going towards the blue, it's not, it's like not very related to the cancer. So using this clustering method, we can, in the first phase, we can identify whether it's a cancerous or non-cancerous. And in the second stage, we can also sub-classify as the NLSC. LC means non non non-cell lung cancer, non small cell lung cancer, or small cell lung cancer. And then the normal patients are also there. So the same criteria applies here also. The yellow samples are like more more responsible for the cancer, and the blues are the less responsible for the cancer. So based upon this, we can identify certain microRNAs which are taking participate into the cancer, and based upon that, we can do the further cancer diagnosis thing, and we can also develop our own um, um, our own panel for the lung cancer diagnosis. So with the first experiment, we did the second experiment on the selected microRNAs. So now out of these many microRNAs, we have selected only these microRNAs, and based upon their performance for the contribution to the lung cancer and small cell lung cancer, we can ultimately identify which are the microRNAs are taking part into the cancer thing. Correct. Again, so ultimately, what we have discovered out of this analysis, that initially there were 73 microRNAs, and ultimately we have concluded that the 10 microRNAs which are identified as the potential for the lung tumor. Correct. So ultimately, what we have to do, we have to focus on only these 10 microRNAs. So if I compare this kind of problem, this kind of solution with the journal data science thing, so you can correlate this problem or this kind of application with the feature selection and feature reduction thing, but very biological, very biological. So maybe these kind of techniques are not very useful in the journal application, and vice versa. Also, the application, the techniques we use in the journal application are not be very useful here. But also, just to understand, you can say it's a kind of selecting appropriate microRNAs who are contributing for the lung cancer in the small cell lung cancer and non-small cell lung cancer patients. So it's like this. In one another case study, I'll show you how we develop drug development system in any patient or any potential patient using the genomics data. So it's preclinical research is the part what I have shown you earlier up to the microRNA selection. So after this, the clinical trial happens. We perform on a bigger population, and once clinical trials are successful, then we go for the approval. So every country, every continent have different kind of medical bodies who approve any kind of solution into it. You must be aware about the clinical trials for any particular medicine. Like if you give solution for the even for the fever, you have to go through of the clinical trial, and once clinical trial is successful, then only you can sell it into the market. So the main thing is FDA into it. FDA is the approval body from USA. So if FDA is approving it, it means your drug or your medicine or your treatment is good enough for the normal population, and you can come up with with that kind of solution. So this is kind of that thing. So this thing is we have developed deep learning based for the cancer patient stratification in the multi-omics data, like different type of cancer. Like if we the previous application we just did with the lung cancer, but this is for the multiple cancer data. uh, multiple cancer data. So it's a same thing. We consider the genetic expression, gene expression, and other indicators or other biomarkers, and then we apply some kind of pre-processing into that data, and then with the help of the appropriate deep learning technique into it, and the molecular pattern analysis techniques, we can perform different uh, uh, we can perform different kind of statistical analysis for the different application in the multi-omics data. We can also do the stratification. We can also do the diagnosis thing. We can also use that information for further research. So we can do every kind of thing into it. So this framework has been developed. So ultimately, with the molecular pattern, we can do the clustering, we can do the subtype analysis of any cancer. We can also see the effect of any particular disease into the patient. It's all dependent on the what kind of molecular pattern are being generated with the help of the deep learning. So this is also one application. So I must tell you, this is not a very difficult thing. We have used a simple deep learning technique which is called ResNet 50. So it has 50 hidden layers, and um, uh, uh, based upon the learning of the weights of those hidden layers, this kind of data or this kind of pattern can be generated. On also, nowadays generative AI and transformer models are also becoming very popular. So those kind of models can also be utilized into it. So I would say, if you are if you are a uh, 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 computer science engineering having a good knowledge of data science but a limited knowledge of the biology things, that of course you can learn. It's nothing very difficult. So you can also be a bioinformatician or a biomedical data scientist out of that. So this is what I wanted to add one more thing. So then after this, we can also perform the patient stratification, like patient classification into it, and then we perform the classification stratification using the AUC, area under the curve. And as you can see, with the MAUI method, we have almost achieved more than 98% of the AUC, which is quite good thing. So these results are already in patent process, and also in the development process of a product. So we are also coming up with the multi-omic data processing solution for the direct hospitals also, and for the other pharmaceutical companies also. So this is one area in which we are also focusing much. So these are the conclusions from our previous study, that more we, because it's a very new technology. So more we start doing the sequencing of different biofluids, the price will automatically come down. More players will come into the market, and like job related to the this kind of work will also be came into the came into the picture. So these are the main takeaway from that. So I also wanted, I desperately added this slide into it. So as a computer science engineer or a data scientist, you have uh, you can contribute at each and every step of this solution development. Like you can be a main member of the data collection team, because as an data scientist, you know how to preserve the data, how to collect the data, what type of information to be collected from the data, and also you can perform the pre-processing part. Then data analysis and visualization, that how, what kind of data is it, the initial visualization